Hello students, welcome to EPG Pathshala. I am Dr. Rajiv Jain from Jivaji University Gwalior. Today we are going to discuss about a module Biomineralization under paper Environmental Biotechnology. Here in this module our learning objectives will be what is biomineralization, different modes of biomineralization, organisms for mineralization, mechanisms for biomineralization, biochemical reactions involved in biomineralization, biomineralization of metals, and significance of biometallurgy. All these things we shall study in this module. So why the need for biomineralization and what is mineralization? So for that purpose, I want to emphasize here that one of the aspect is that how minerals are taken out from the earth crust. Mining is one of the humankind's oldest activities. By mining, we mine out different types of products, whether it is of metals, gold, different types of our ores, alloys, all are taken out by mining. Our coal for energy purposes we take out by mining. It is one of the oldest and first known industrial activity which is used to extract metals from the mineral ore. Metals are extracted. In the ancient time also, gold is extracted from ore. Different types of metals, useful metal, copper, which is very useful, is extracted from different types of ore, and these ores are taken out by mining, and this mining activity causes different types of adverse environmental effects. Ores are chemical compounds of different metals occurring in nature and are known as minerals. These minerals exist in the form of oxides and sulfides. The conventional mining involves following steps. Step 1 is the digging of the ore from the earth. After that, crushing of the vast quantities of ore from the potential sites and its grinding. Then smelting of the pulverized ore, which is highly polluting and energy intensive. So, due to this mining activity, which I explained you earlier, air pollution is also caused. How? Air pollution is caused due to the release of particulates in the form of dust during excavation, digging, shoveling, blasting, or processing of minerals. Water pollution is also caused by use of acids, alkalis, or other chemicals in beneficiation of ore, leading to the formation of wastewater. Means here, due to mining activities which involve number of steps, it causes air pollution as well as water pollution, because we take out ore, we take out ores from the earth by digging, different types of operations. Then different types of operations are done here: smelting, roasting, purification. So different chemicals are added. So they cause both type of pollution. Different types of oxides form. They cause air pollution. And for treatment, we add different types of chemicals, different types of things goes into water bodies, and they cause water pollution. Similarly, noise pollution is also caused by different types of mining activities. Here you will see the noise pollution is caused by blasting. If blasting is done by blasting, drilling, and the use of compressed air in metallurgical operations, creating noise pollution. These environmental hazards are only a few significant areas out of several others which demand immediate action. What is biomineralization? For understanding mineralization, we can say biomineralization is the process by which microorganisms mediate and catalyze inorganic reactions to form new mineral assemblages. The goal of biomineralization is to control microbial processes and their environment so as to sequester and immobilize specific heavy metals with a geological time scale. As it has been shown in figure 1, this is an overview of biomineralization process. It is remarkably a widespread phenomenon where bacteria, algae, plants, invertebrates, and vertebrates all synthesize large parts. Biomineralized structures include spicules, ossicles, shells, teeth, spines, and bones. Biominerals is probably magnetite, which has been found in a diverse group of organisms as salmon, humans, and magnetotactic bacteria. Biogenic magnetite can form as a result of either biologically induced production or biologically controlled production. Biologically induced production occurs as a byproduct of an organism's metabolic process. Iron reducing bacteria dump reduced iron into their surroundings where it can react to form magnetite. The minerals formed by this process are in general quite similar to a biogenic magnetite. The biologically controlled production occurs in an organic matrix within a cell. The organism had tight control over the structure and composition of the products, which are called magnetosomes. The magnetite crystals employed by magnetotactic bacteria as well as those in higher organisms are the result of biologically controlled production. Biomineralization is a process by which organisms form minerals, that is, converting an element into its insoluble form. It is biologically induced minerals formed as a result of metabolic activity. For example, pyrite as a result of sulfate reducing bacteria. It is biologically controlled minerals formed under genetic control. The size, shape, and composition are essential to its function. For example, magnetotactic bacteria form bones. In figure 2, process of biomineralization has been depicted, and you can see very well how biomineralization is taking place. Types of biomineralization and modes of biomineralization. There are two fundamentally different modes of biomineralization. In the first one is called biologically induced mineralization, in which an organism modified its local microenvironment, creating suitable conditions for the chemical precipitation of extracellular phases. In the second mode, which is called as boundary organized biomineralization, in which inorganic particles are grown within or on some organic matrix produced by the organism, which has been in figure 3, a mode for biomineralization has been depicted. Bacteria that produces mineral phases biomineralization do not strictly control the crystallization process, resulting in particles with no unique morphology and a broad particle size distribution. Non-magnetotactic dissimilarity, iron-reducing and sulfate-reducing bacteria produce magnetite, siderite, vivenite, and iron sulfide by BIM processes. For example, iron-reducing bacterium Geobacter metalli reducens is a non-magnetotactic anaerobic bacteria that couples the oxidation of organic matter to the reduction of ferric iron, inducing the extracellular precipitation of the fine grain as a byproduct. In figure 4, a schematic representation of the biomineralization process, including the nucleation, growth, self-assembly, and aggregation, has been depicted. So, different types of organisms which are used for mineralization. How to solve this problem of pollution? How to mineralize them? Mineralize means to convert them into their fundamental products, means to convert them into carbon dioxide and water. A variety of different organisms have shown the ability to accumulate an array of different metals. Lead and cadmium have been effectively removed from solutions by species of brown algae, such as Ascophyllum and Sargosura. Means these algae are being used for the recovery of metals which are very toxic, which are harmful to man and animals, but they can be fruitfully recovered. Mycelia of the fungus Rhizopus and Absidicia have been shown to be excellent bioabsorbents. Very bioabsorbent, they are being used as bioabsorbents or adsorbents for lead, cadmium, copper, zinc and cobalt, gold, cadmium, chromium, iron and manganese. All can be taken out by these different types of species involving bacillus species are being used. How what these species are doing? These species are being used as adsorbent, bioadsorbent. They are called as bioadsorbent because these are the living species, so they are called as bioadsorbent, and they are adsorbed there, and they are being concentrated over bioadsorbent from which they can be fruitfully recovered. There are many more types of fungi, algae, yeast, and bacteria that have the same ability to sequester metal ions. Some microbes extract the largest mineral and must themselves be processed to purify it. Some devour the elements compounded in the ore, leaving the mineral behind. Some excrete substances that leach the mineral from its matrix. Compared with conventional extraction technologies, bioprocessing is slow, but it is cheap and natural, clean, and sure, and we can identify the same thing from there. We can extract the same thing from there without polluting the environment. A number of genera of bacteria have been shown to be important in biomineralization, and biomineralization are Acidothiophilus, Leptospirillum, Acidiphilium, Sulfobacillus, Ferroplasma, Sulfolobus, and Acidinias. All these species are effective in removing different types of metals, and so the process is called as biomineralization. If we are not using bacteria or algae or fungi, then it is mineralization. But if we are using some living being, then it is biomineralization. Mechanisms for biomineralization. Different metals can be dissolved from insoluble minerals either directly by the metabolism of microorganisms or indirectly by the products of their metabolism. The biological reactions involved in extractive metallurgy are primarily oxidation of sulfur or mineral sulfides. Generalized reaction often used to express the biological oxidation of mineral sulfide involved the reaction where the where M is the bivalent metal ion. The mineral sulfide serve as an energy source in the presence of other nutrient, or we can say here the sulfide of mineral is serving as the energy source in the presence of various other nutrients present therein. Some of the chemolithotropics microorganisms are capable of direct oxidative attack on mineral sulfides. It has been proved by electron beaming studies that the bacteria attach themselves to the surface of sulfide mineral in solutions supplemented with nutrients. Bacteria dissolve sulfide surface of the crystal by means of cell contact. The cell of living organisms also been found to attach with hydroxides of metals in solution. Insoluble sulfide minerals can be biotransformed by microorganisms in absence of ferric iron under conditions that preclude any likely involvement of ferrous-ferric cycle. Although many aspects of the direct attack by bacteria on mineral sulfides remain unknown, it is apparent that specific iron sulfide oxidizers might be playing an important role. Microbial involvement influenced by the chemical nature of both aqueous and solid crystal phase. Bacteria appear to attach specifically to the sulfide moiety of mineral rock surfaces, which are the regions that contain the energy supply for the bacteria. Attachment and sulfide oxidation results in pitting of the mineral surface. The extent of surface corrosion varies from crystal to crystal and is related to orientation of the mineral. The metabolite that aids the dissolution of mineral sulfide may also be produced. The substance has been postulated to act either by oxidation of iron 2, stabilization of molecular sulfur on the surface crystals, and direct attack on crystallized mineral sulfide surfaces. As shown in figure 5, that biologically controlled and biologically induced mineralization is taking place and different types of products are being formed in direct mechanism. The role of ferric sulfate and oxygen has been cited in the oxidation of mineral sulfides. Minerals such as galena, chalcopyrite, bornite, and sulfurigide are oxidized more rapidly than pyrite and marcasite, whereas covellite, chalcolite, and molybdenite are oxidized more slowly. Ferric iron, either or in combination, is the most important chemical species involved in the indirect attack on the sulfide minerals. Stage 1 involves chemical leaching of ore by iron 3 from iron 2, whereas stage 2 involves chemical leaching of ore by iron 3 in acid solution. This reaction involved in indirect mechanism can be represented as given below, that with the bacteria converts iron 2 to iron 3, and now this metallic sulfide iron 3 is again reduced to iron 2 and it forms metal in the lower oxidized form in acid solution. In the presence of iron oxidizing bacteria, the ferrous ion produced by these reactions can be oxidized to ferric ion, thereby establishing a cyclic process. In aerobic process, the ferric sulfate generates ferrous sulfate and elemental sulfur from pyrite. Biomineralization results from interaction of extracted metabolic products such as iron, gases, polypeptides, electrons with extraneous metal ions in the surrounding environment. Now we shall discuss biochemical reactions involved in biomineralization. The general physiology and metabolism of iron oxidizing bacteria, thiobacillus, is well documented as compared to others, and the organisms belonging to this group have more potential for commercial applications. Further, the specific growth conditions for the organisms, especially low pH, avoid the cross-contamination. The bioleaching of metals is related with mode of metabolism, which is governed with physiological process of the organism. This metabolism incorporates the oxidation of suitable substrate and is carried out at the expense of certain nutrients taken into the bacteria in support of the growth. The energy of source of the ferrooxidants is reduced iron, which is stable in acid solution. The following biochemical reaction occurs for the control of the physiology of the organism, where iron 2 is converted to iron 3 and electrons are released. The characteristic reducing potential is available for the reduction of molecular oxygen through the following mechanism, where oxygen reacts with proton and forms water molecule. The formation of ATP through phosphorylation is governed by the reaction where ADP plus IP and it forms ATP. The pair of electrons transferred yields enough energy to ensure ATP formation. Thus, chemical energy is converted by oxidative phosphorylation, the biochemical equivalent of respiration to ATP that is universally recognized as the form of metabolic energy which the cell utilizes for transporting substrate and nutrients into the cell and metabolic products out of the cell, vibration and locomotion, and for biosynthesis. ATP is hydrolyzed to ADP and inorganic phosphates and releases the energy. Two other important reactions for autotrophic growth, the formation of reducing power and fixation of carbon dioxide, which are dependent upon the energy generating processes of metabolism. The reduction of pyridine, NADH, NADPH provides the organism with reducing power and is generated from the oxidation of ferrous iron. Energy is also required to move the electrons against a redox potential gradient cytochrome to the NADH, where following reaction takes place. NAD takes up 2 electrons and protons in the presence of ATP and it forms NADH and ADP is formed. Then this formed NADH reacts with proton and the NADP plus and forms NADPH and protons are released as shown in the reaction. The carbon dioxide fixation reaction in T ferrooxidants is linked with the production of energy from iron 2 plus oxidation. The pathway for carbon dioxide assimilation is a reductive pentose phosphate cycle that includes two characteristic enzymatic reactions. Number one enzymatic reaction, phosphorylation of ribulose 5-phosphate catalyzed by phosphororibulose kinase to yield ribulose 1,5-diphosphate, and the second reaction involves ribulose biphosphate carboxylate, which catalyzes a reaction between one molecule of carbon dioxide and one molecule of ribulose 1,5-diphosphate with a concomitant conversion to yield 2 molecules of 3-phosphoglyceric acid. Laboratory studies have also indicated that the iron-grown cultures can be adopted to use organic substrates for energy and carbon under specific conditions. However, little is known about the capacity of organisms for heterotrophy and or mixotrophy in nature. Like the biochemistry of iron oxidation, the biochemistry and physiology of inorganic sulfur oxidation by thiobacillus is well known. The mechanism of sulfur oxidation is similar to that of thiobacilli. Elemental sulfur, tetrathionate, thiosulfate, and sulfide support the growth of organisms. In all the cases, sulfide is the key intermediate molecule. Energy is produced from oxidation by the use of enzyme, sulfide oxidase, EDP, sulfurylase, APS reductase, and adenylate kinase. An alternative oxidation mechanism for sulfur has been proposed in which elemental sulfur is oxidized to sulfide and then to sulfate by T ferrooxidase through ferric ion system. So, if I say that large amount of heavy metals are released into the environment each year from industrial waste, water through inefficiencies built into the technological activities used directly in the processing of metals. The metallic species released into the environment tend to be maintained there indefinitely, circulating and eventually accumulating through the food chain, posing a serious threat to the environment and public health. There are numerous methods currently applied to try to remove and recover the metals polluting our environment. These include such techniques as chemical oxidation and reduction, membrane separation, liquid extraction, carbon adsorption, ion exchange, electrolytic treatment, electroprecipitation, coagulation, flotation, evaporation, hydroxide and sulfide precipitation, nitrification, and crystallization. These methods differ in their effectiveness and cost, also often tend to treat the metal totally as waste without the possibility of recycling. One potential alternative to existing technologies for removing heavy metals from industrial wastewater is bioabsorption by microbial cells. It is well known that the metal ions are involved in all aspects of microbial life. A small concentration of metal ions are needed within microorganisms to stabilize a range of biological structures such as enzyme cofactor and serve many other functions. When essential metals are at high concentrations, however, they become toxic to microorganisms. In order to deal with the toxicity, some microorganisms have developed mechanisms, including one by which they convert the metal ions to soluble metal complexes. These metal complexes form as precipitates external to the cytoplasm of the microorganism, rendering the metal harmless. This normal biological process may be potentially effective procedure for solving and controlling major industrial environmental problems associated with metals, including biomining of dilute solutions found in pit mine lakes. So, what is the significance? The biometallurgy is of significance due to the following advantages. Its wide application for the recovery of valuable metals from low grade ores, the relative absence of air, water, and land pollution, low energy requirements and operating costs as compared to conventional processes for recovery of metals from low grade work, the degradation of a variety of mineral forms, selective leaching possible, one metal solubilized with the other remains insoluble. Application most useful is desulfurization of coal for burning is free of sulfur and further pollution problem is avoidable. Suitable for less developed countries as it eliminates the need for some costly important heavy metal equipment. In the last, I shall discuss here advantages and disadvantages of biomineralization. Using mostly recyclable biomaterials, we can extract minerals from low-grade ores without the need of large ovens, cookers, electricity, etc. The machinery doing the work are self-reproducing and self-regulating. Therefore, a very long-term system could be established that runs well constantly and independently. It is easy to imagine a mining system that does not require local human intervention at all. The bacteria and this methodology of mining currently exist and will only be improved as terrestrial and non-terrestrial mining requirements drive genetic manipulation and biological engineering. This method can be used with low-grade ores. There are some disadvantages also of biomineralization. One of the disadvantages is that the balance necessary to maintain sufficiently high yields might be difficult to impossible without significant control mechanisms. Overall, a small material can be harvested in short time. However, given extensive time, significant amount of material can be extracted. For mineral extraction and for possible extraction of inorganic materials from regolith, it may be necessary to process large amount of material to gain small yields. However, availability of suitable regolith is extremely high. The bacteria could be very susceptible to environment, and if they all die, all the remaining machinery is mostly useless. Therefore, dozens of backup culture have to be maintained at a very high expense. So, students, let us summarize what we have learned in this module. In this module, we have learned bioleaching of minerals. It is one of the bioleaching and biomineralization. It is one of the oldest thing to know how to recover. नहीं दोबारा से बोलूंगा repeat. In the present module, we have learned what is biomineralization, how biomineralization is carried out, how different types of metals from waste are recovered, how different types of waste can be recovered by using different types of microbes, different types of algae, fungi, bacteria, and also by using different types of chemical processes. Thank you.